The evidence is in ways that Tourette's kids are not neurotypical. That's a word that people toss around a lot today. Tourette's kids might be even more competent than their non-Tourette's. Of course, Tourette's, unfortunately, comes frequently as a package with OCD and ADHD and sometimes even autism. So these things in general are unlikely, although OCD sometimes makes people a bit more competent if they choose the right careers and right things to do. But I think we can say with confidence that there's no real deficit in Tourette syndrome. There's behavior that disturbs the people they interact with and sometimes the kids who are affected, but there's no reason that they shouldn't lead highly productive lives.
Welcome to The Uptick, brought to you by the New Jersey Center for Tourette Syndrome and Associated Disorders, empowering children and adults through education, advocacy, and research by sharing the stories and experiences relevant to the TS community. Welcome to The Uptick. I'm thrilled to be speaking with Drs. Jay and Max Tischfield. They are Tourette researchers at Rutgers University, and they work for the world's largest genetics sharing repository for Tourette syndrome. The repository was founded in 2007 in partnership with the New Jersey Center for Tourette Syndrome as a way for researchers around the world to gather and study genetic data on Tourette patients and their families, and also genetic info on other conditions. The repository is recognized and funded by the National Institute of Mental Health, and through its work, we've been able to identify gene mutations that are thought to be implicated in Tourette syndrome. So they're doing some really phenomenal, groundbreaking work here. I want to start with the biggest, most important question I have. You are a father and a son, both involved with Tourette research. So I'm dying to know, in your family, what are the dinner table conversations like?
We just tick.
No, it's present in our family, but neither of us have a diagnosis. My DNA is part of that large collection you talk about. And within that study, I think I'm coded as a partially affected individual, meaning that I have some traits that you might find in someone in Tourette's, like repetitive motor behaviors to some degree, but I don't actually have a diagnosis.
What got you both interested in Tourette research?
I wasn't interested in Tourette research until perhaps 20 years ago. I was doing other things, and I didn't know that much about Tourette's, except I had an affected son. Max is not our only child, or he acts like it. Then this woman called me, and that was Faith Rice. She said, "Are you doing Tourette's research?" And I said, "No." And she said, "Why not?" I said, "I don't have the slightest idea why I'm not doing anything." She said, "Well, you have to do Tourette's research." She said, "I'm coming to your office, and I'm bringing a whole bunch of families with me. We're going to talk you into doing it." And she talked me into it, and I went to one of the galas right after that. And there's another one, by the way, next year. And Tim Howard was there. And I met Tim, and I don't know, I just got roped into it. And it was a smart move. Most of the time when I do smart things, it's because other people tell me to do them. We decided how we could start. And the main issue was a lack of human samples. There were none available anywhere, not in the National Institute of Mental Health repository, which I ran, so I knew that, and not elsewhere. The few that had been collected by what used to be the TIA, the national organization, were not available to other people. They were used by some researchers. So we decided to start the NJCTS Sharing Repository. And a few years later, we were quite successful. We gathered together a lot of small, meaning children and parents, and a few large families, including grandparents and some other relatives. It was impressive enough for us to get federal funding at that point. So we applied, and that's how I got interested in Tourette's. And then Max came along to Rutgers. I guess I was Faith Rice to him. I said, "Why aren't you doing Tourette's research? It's a good disease, and you can get funded and accomplish something." And the rest is history. Is that fairly correct, Max?
Yeah, I wouldn't call it necessarily a good disease. I think that Tourette's is a disorder that, compared to its cousin, autism, there's really a lot less known. there's a paucity of basic clinical.
That makes it good. There's lots of room for researchers to discover stuff. There's only about 1,000 people doing autism research now because there's so much money in it. There was no money in Tourette's. Let me just say, I think Tourette's is a good disease to study. I obviously don't mean a good disease to have, because not only does it have a psychiatric component, but most physicians would say the movement component is the key feature. And movement is something you can see. It's much easier to diagnose than the autism syndrome traits.
Yeah. So when I was looking for an independent faculty position in 2017, I was intrigued by the idea that with the discovery of the first high confidence gene mutations linked to the Tourette disorder, we could use DNA editing technology to introduce these mutations into a mouse model, thereby generate the first ever mouse models that actually harbored a mutation found in a human. And the question then became, can we use these mice to actually model the underpinnings of Tourette disorder? And can we really start to break open or illuminate this black box that had really eluded researchers for a long time? And just a little bit more about my background, and I think this would underscore why this was a mission that I really wanted to take. In addition to having Tourette's in our family, my training, so I was an undergraduate at Rutgers. I majored in cell biology and neuroscience. And then I went on to train in Harvard's program in neurobiology. In particular, I trained with a neurologist named Elizabeth Engel, and she was involved in basically characterizing rare neurological disorders. So these were disorders that were not present in the literature, or if they were, there was no known cause. Really, my training then became to understand how do we take a gene mutation that we find in a human, put that into a mouse, and actually model the key features of that syndrome, so then we can describe that to clinicians, and they can then better diagnose and treat their patients.
I had a secondary interest. Max is a neuroscientist. I'm a geneticist, and I've worked with NJCTS at the Tim Howard Academy and what used to be called Camp Bernie and at various events. We just had one maybe a month ago, a meet and greet with families to talk about current research. And the question I'm asked most frequently, and I don't have an answer, is what medications are going to be most effective. Nobody has developed any drugs specifically for Tourette's. And I thought if there were a good animal model and a person like Max could show that, then we would have an animal to test drugs on, and we could really rapidly screen either known compounds or new compounds for their effect on Tourette's-like symptoms in mice, and then go to humans.
It's interesting. I mean, you're both Dr. Tischfield, so I'm going to call you Dr. J versus Dr. Max, just so we're clear here. Okay, I want to be respectful.
Plays better basketball than me.
I like that. Well, Jay, I remember you telling me, I asked you at the Academy a couple years back, as a result of the research, how many unique genetic profiles have we found that can lead to Tourette syndrome? And if I recall, you said it was like 500 or something, like a large number.
Well, this number we found and the number that we statistically proposed based on what we found. The 500 is the latter number.
Okay.
We think there could be as many. We have about a half dozen at this point, or if we count the old literature, it might even be a dozen genes, with 3 or 4 being very high confidence. The one that Max is working on, this so-called CELSR3, is a very high confidence gene.
The challenge I see is if people with Tourette, there's different genetic chemistries underneath that, how do we find and prioritize one that could lead to a drug that helps the most people? Do we find that, like, yes, there's a lot of genetic diversity under Tourette, but people tend to cluster into these, okay, this gene gets mutated a lot, this one gets mutated a lot. How easy is that? How complex is that?
Tourette's, from the point of a clinician, has a common diagnosis of motor and verbal tics. So all of these different genetic causes may have one or several common intersections. If one can target these intersections with a drug, then you could have a drug effective with different kinds of Tourette's.
But I will add that there's a challenge to this. We believe that these are mutations that are associated with the disorder. We gain confidence when we sequence the DNA of these hundreds of individuals. And in some cases, what we'll discover is that there are changes in a very specific gene that one individual has that another unrelated individual has. So when you start finding multiple unrelated individuals and they all have a distinct change in the same gene, it doesn't necessarily have to be the same mutation. If it is, that's great evidence. But if they have mutations, changes in the same gene, and they're unrelated, and if their parents who are unaffected also do not have that change, so it arose spontaneously in the individual with Tourette's, we gain confidence that we have a good gene candidate. That's why we start using the term high confidence. The false discovery rate is such that we believe that this gene is indeed associated with the disorder. Now, that's great. We get excited when we find those genes, but we now have the next challenge. And that is, how do we really prove that these genes are indeed linked to Tourette's? Can we do that in an animal model? That is potentially fraught with difficulties. For example, we're not quite sure if mice, which is the model organism that we typically use, if they actually tick like a human. What is a tick in an animal? And we don't have a good definition yet of what that is. So what we tend to do is we take these genes of interest. Let's say we have a gene that Jay mentioned, CELSR-R3, and we have 9 different individuals in our study that have a unique mutation in that gene. And what we've done is we've selected a subset of those mutations, and we've used a DNA editing technology called CRISPR-Cas9. Many have heard of this. It's won the Nobel Prize a couple years ago. It's now being used for gene therapy. And we CRISPR, where we edit the genome of these mice and we put these different mutations in.
Unfortunately, the genes in mice and humans are almost identical. It becomes an easier task from that point of view. I would add that while not looking, if we saw something like a human tick, we'd be ecstatic. But assuming we're not going to see something like that, we look at all the behavioral parameters of the mice. And Max will say more about that. But we look at all the parameters to see how they differ from their non-mutant litter mates.
And that's a challenge. How do you pick that parameter? How do you pick a test? So we have this mouse, this quote unquote Tourette-like mouse. What are we going to do with this mouse? And I will tell you that when you look at the mouse just with your naked eye, you don't see much, right? And that's typical of many individuals with Tourette's. You don't notice it at first glance, right? Sometimes you really have to pay attention. Tics wax and wane. And in these mice, how are we going to approach this? For example, we could look at general levels of activity with the idea that potentially the hyperactivity in some forms of Tourette's might be present in these animals. We can look for changes to motor behaviors that might be repetitive-like, such, for example, mice like to rear on their back hind limbs, so they'll rear up in the air. And that tends to be one domain that we actually look at and actually is increased in our mice.
But you have to do it very quantitatively. You can't just sit around and say, oh, these mice, you have to have a mechanism.
Yeah.
And we do that using AI learning programs.
Can you elaborate on what tics in mice look like?
Yeah, this is a really important question. I would say, again, we don't have any basis or any real strict criteria that we can use to find a tic. And that really brings up problems in the literature. And we've encountered this ourselves. We have to be very careful how we describe some of the motor behaviors. Those that are very close to the field of Tourette's will argue that what you find, you can't really call a tic. You have to just describe it for what it is. Whereas other people in the literature, you'll see commonly the phrase tic-like stereotypy. So
that's how they cushion it. Yeah.
Right. Or I would say that there's one mouse out there that has been around for a long time. And it's a mouse that they basically played around with the dopamine signaling in a way that's very artificial. They did something that you would never find in a human. It's supposed to basically mimic higher states of dopamine in certain parts of the brain. And that mouse has probably the most tic-like movements of any mouse out there. But the problem is it doesn't actually resemble in any way any type of DNA change or a change that we can say this is what a person with Tourette's actually has.
And that particular change has never been observed in humans, just hypothesized.
This mouse, this particular one called the D1C-T7 mouse, they just found a way to activate the dopamine type 1 receptor in the forebrain. And that mouse has a lot of interesting phenotypes and does very much look like it tics. But it's problematic in that we can't trust the underlying change has anything to really do with what you would see in a human. Now, one thing I will add, there is a behavior that has been documented in humans that you can study in mice. And this is something called pre-pulse inhibition. And this is a test in which you basically startle a mouse with a loud sound. It could be a sound or a tactile stimulus. It processes that initial startle, and then you startle it again. And what the mouse will do is it'll jump in the beginning to that initial startle. If I clap my hands in front of you really quickly and you're like, whoa, and you jump up. But let's say 10 seconds later, I clap my hands in front of you again. You've kind of habituated to that stimulus. So you don't jump as high or you're not as surprised. And that's exactly what we test in our mice. And individuals with Tourette's in human studies have been shown to lack the ability to properly filter or process that first stimulus. It's called a sensory motor gating deficit. Sure enough, we did this behavioral paradigm in our mouse models because it's very similar to what you would do in a human. And very interestingly, our mice, almost all of the models we've made thus far, both males and females, show a deficit in pre-pulse inhibition. We propose, based upon our mouse models, that looking for sensory motor gating deficits could be a core behavioral finding in a mouse model. With that, you could ask, does it respond to drugs that you would prescribe to a subject that has Tourette's? Back in the day, it used to be haloperidol. These are drugs that work on dopamine. And what we're finding is, indeed, the drugs that are used in humans are actually showing effects in mice, and especially in these sensory motor gating deficit assays.
Which is very encouraging based on what I said before about drug development.
These results should all be out early next year. And I think it just goes to show that the goodwill of those who donated their DNA into this DNA cell repository has led to tangible results that we were able to then go into a mouse, look at these mutations and say, wow, we may have the first real valid models of a Tourette-like mouse. We may have finally generated the first one.
Yeah, I think Max even understates the case. I have a longer perspective due to age. There are people walking around that have the exact same mutation as our mice, or It's actually the other way around. The mice have the exact same mutation we put it in as the humans. We want to take it to the next step. If we have enough people, and we really do need more, we can start finding more genes and looking for those common intersections where the gene effects might meet. I don't think there are any other large groups funded for Tourette's. We are the only ones in the federal government. And we have to convince them to give us another 10 or 20 million bucks.
But the neat thing is, once it's processed, that genetic data is available online for researchers all over the world as part of this collaborative.
Not just the data, the actual samples.
Oh, they can get the samples too.
Oh, yes.
Why might they want to do that as opposed to just having it sequenced?
Let me just say we've done more than make mice. We've also made what are called iPSC, induced pluripotent stem cells. And you read a lot about stem cells today. We're also studying these, and the stem cells, we use the same kind of CRISPR technology, or we use the blood of the patients, depending on the circumstance or where it came from and such. Anyhow, we have this collection of stem cells that also models all the mutations that we've collected in Tourette's. So we have a mouse and a human resource, and of course, our subjects that can contribute to this research.
And I want to give our listeners some info on that. I participated in it a number of years ago, so you should still have my sample.
It's all anonymized. I don't know which sample is yours.
I'm in there somewhere. My trio is, because actually my whole family did it, because I have 2 of my 3 siblings also have an active Tourette diagnosis, and we believe it came through my mom's side. But I did the blood sample. We each filled out a lengthy questionnaire about all of our tics and co-occurring conditions, family background. They were looking for a lot of different variables to understand. I think there was a meeting with Dr. King to discuss the process. He kind of managed that and what would happen next. And that was it. I filled out the form, had the interview, the blood was drawn. And then, the highlight for me was really every year at the Tim Howard Academy, we take the students through the repository. So they actually get to see the lab where this data gets worked on. that was really neat and really made a lot of this come to life.
Next year, it will be super because we've actually finished construction of the new repository, which is many times larger than the old one. And people were impressed by the size of the old one.
Oh, I'm looking forward to that. What would you say are some common misconceptions or just assumptions we had about Tourette genetics that your research has helped clarify, demystify, explore?
There are a lot of social issues, as you well know, surrounding Tourette's and how society treats people with Tourette's. And there were questions. I think they've been well settled now as to whether or not people who have Tourette's are less confident. Tim Howard said in his book, and said to me personally, that he always felt he reacted quicker because he had Tourette's. I don't know if that's true. No one's like Tim Howard.
He's a special guy. Yeah.
Yeah. No, he's just like 62 feet or something. But we've seen a very large number of Tourette subjects now, and we've actually made mice. The mice are easily as competent. In fact, they may be quicker learners of certain things. And Max can talk about that in terms of his habit formation theory of Tourette's. The evidence is in ways that Tourette's kids are not neurotypical. That's a word that people toss around a lot today. Tourette's kids, maybe some of them might be even more competent than their non-Tourette's. Of course, Tourette's, unfortunately, comes frequently as a package with OCD and ADHD and sometimes even autism. So these things in general are unlikely, although OCD sometimes makes people a bit more competent if they choose the right careers and right things to do. But I think we can say with confidence that there's no real deficit in Tourette syndrome. There's behavior that disturbs the people they interact with and sometimes the kids who are affected, but there's no reason that they shouldn't lead highly productive lives.
Tourette in one individual is not going to be the same as Tourette's in another individual.
Yeah.
And any type of spectrum disorder, that's what we call these disorders, right? Where there's a whole spectrum of different types of symptoms, whether it might be associated OCD, ADHD. I mean, the list goes on. Conduct disorders, whether there might be higher incidence of substance abuse. There's really a large gamut. And we can't necessarily put everyone into the same box. And as a researcher, it's very important to understand that, because when we read the literature and the literature says Tourette's is caused by this, based upon this study with 30 individuals, you have to take that with a grain of salt. And oftentimes these studies, clinicians are trying their best, but they're based on a limited sample number. In some cases, they're not reproduced. But yet these studies kind of set the standard or the basis for what we think is Tourette's and what we think causes Tourette's. We want to take everyone's findings, we want to take them seriously and test them. But in some cases, I think it's important to realize what we think we know about Tourette's is based upon imperfect models, maybe imperfect experiments. The hope is over time that we can develop these better models, and with tools through artificial intelligence, can we harness these new technologies with these better models to gain a better representation of what Tourette might be in terms of brain changes?
We've been using these AI machine learning models on the mice to characterize their movements. And now we have an engineering student who's working on it with us. We'd like to do the same thing with humans.
Wow. Yeah. So, because it sounds like even if these mice have that, we give them that genetic mutation that some people with Tourette have, the manifestation of it isn't going to be the same. They're not going to get the tics as we do. So, but yeah, and we still study it in them. How does family history influence the likelihood of developing Tourette? So that, Jay, this may be more your wheelhouse, the genetics of Tourette. What are some patterns and trends we've observed in the genetic data?
Remember that in our current collection, everything is skewed toward new mutations. We look for trios where there's no family history, where there is only one affected SIP. And that increases the likelihood that that child is a consequence of a new mutation, because when we look at the parents, we don't find the mutation that the child has. We think perhaps about 15% of the Tourette's cases are a consequence of new mutations. Then you have some well-documented cases. Our colleague Matt State, and I was a co-author, we document a family 25 years ago that had a huge number of cases because of a specific mutation in a particular pathway. So Tourette's in that family was inherited the same way in every person.
Wow.
We have published in what's called a polygenic risk score, PRS for short. That looks at all the genetic background, all the mutations, all the variants of all the genes that we can test, as many as 18, 20,000. And it calculates a score based on what we see in Tourette's patients. And that score has useful but minimal predictive ability when we look at somebody whose diagnosis we don't know. There are at least three kinds of genetic Tourette's. There are new mutations, there are inherited mutations, and there are lots of small mutations which cumulatively cause Tourette's. But it may be gene function can be influenced by environment. This is a field called epigenetics. And epigenetic changes can mimic mutations. They're not inherited generally the same way as mutations, but functionally they mimic mutations. So it could be, and I don't really have an answer, that a large number of Tourette's cases are the result of environmental factors. And we've looked at some of these. Nothing really stands out thus far.
I loved how you broke it down into the three big kind of buckets or clusters. You acquire the mutation from your parents, it's a new mutation, or it's accumulation of many smaller kind of mutations that add up and lead to Tourette.
Yeah.
Are we able to say with any level of specificity, like what percentage of our population falls into each of these categories?
That's a good question. But so let's say 15% of the subjects, patients, are new mutations. And let's say, just round numbers, so I don't have to use my calculator. Let's say 1 in 100 people in the population have Tourette's. So 15% of that 1 in 100, and you can just do the math. I can't say with any certainty what percent are inherited, but my guess is, it's at least 15%. And the small mutations, probably somewhere in that range. So I would say that maybe as close to half of all Tourette's is a consequence of mutation. So 1 in 200 have mutations.
When it comes to the treatment, have we tried giving specific drug candidates that are in the pipeline? Do we give any of these experimental drugs to the mice to see if they can do anything with the mice that we've now imbued with this mutation, this gene?
That's the hope. That's what we would say is the promise, potentially, of a really good model. Now that we have confidence that these mice are expressing what we call Tourette-like behaviors, we can ask how experimental drugs or experimental therapies may work in those mice to mitigate these Tourette-like behaviors. And that's certainly something that we're interested in doing. And going forward, we're in the process of or actively seeking out partnerships with pharmaceutical companies who want to test their drug candidate in our animal models, with the hope that the evidence that you might gain from that study could motivate the onset of a clinical trial, for example, where this drug may then be tried to see its effects on a specific, let's say, subdomain of Tourette's, motor behavior. But that's clearly what we hope to do. But beyond that, I would say that the neuroscientist in me, I was trained as a geneticist originally, but my degree is in neurobiology. I think what we want to do is not just necessarily throw all these drugs at these mice and see what they do, but ask why they're doing that. Therefore, we can start to tailor, better treatments for individuals that might have different types of Tourette's.
And also, let me just say, if you're going to invent the drug from scratch, the process of using it on people is very complicated. You start with animal models, you start with bacteria. You have to show the drug's not poisonous, not mutagenic. What I'm looking for, if I had my way, once Max figures out how these mutations work, I would see if we have any drugs on the shelf that were created for other purposes that might be repurposed for Tourette's. And if we have these drugs on the shelf, and I've been involved not with Tourette's, but with other diseases where this has been a remarkable success, in a disease called progeria, for example. If the drugs have been approved for human use, then it's much easier to go right to humans and do a clinical trial.
I was surprised and honestly disappointed to hear that when we imbue mice with these mutations, that they don't exhibit the tics as we see them, the motor,
You were disappointed. I was hoping
you'd say, oh, they squeak more. Okay, great. Vocal tics. Oh, well, and they do more movements. Okay, great.
Actually, we haven't tested that. Mice squeak at ultrasonic frequencies that humans can't hear.
Oh.
And I just set up a new lab just today, actually. I bought the gadgetry that one needs to listen to mouse vocalizations, and we're actually going to see if they squeak more.
Really? So we hadn't explored that before.
Think about tics for what we perceive them to be, whether it be a motor tic or a vocal tic. But I would really stress that we need to ask, what is the underlying basis of a tic? Not just the tic itself, but what's causing the tic? And can we model that in a mouse? Something I might pose to you, Michael, when you have a tic yourself, do you ever find yourself ticking in a particular time or a particular location or a particular context?
Absolutely. Yeah. Absolutely. Yeah, definitely patterns. And
when you think about a pattern, something you do often, what would you call that?
A habit.
That's right. There are studies in humans that suggest that those with Tourette's may be more habitual, meaning that they may rely more on habits than other types of strategies. And the idea that tics might actually be a maladaptive habit in response to urges and unpleasant sensory stimuli, such that in the beginning, when you learn a tic, I think you could probably sit here and say, yeah, I remember when I learned that tic, or I was around this person and I picked up that tic. You learned it. Now, the same mechanisms as our brains used to learn things, dopamine, for example, these are pathways and neuromodulators that are involved with not only how we learn to do something for a goal, to achieve something, but after a while, if you repeat that enough, it becomes a habit. And habits are elicited unconsciously. They're elicited by sensory cues in your environment. They're elicited by emotions, feelings, something that you associate with something that you've done in the past that has led to a reward. So if you learn a tic in the beginning and you feel an urge and you tic and you get that relief, if you repeat that enough, that becomes habitual. And what we have discovered, and this is actually very exciting, this is where our unpublished research is going, is that our mice are more habitual. Like a study in humans published 7 years ago suggested that patients may rely more on habitual strategies. We have been testing mice. It's a 40-day procedure. You have to sit down with these mice sometimes for 10 to 14 hours a day for 40 days straight. There's no Saturday off.
Wow.
There's no Sunday off. Even going grocery shopping for my students is tough during these assays.
Wow.
But we teach mice to form habits, and then we teach them to suppress their habits. Tics are hard to suppress. You can do it, right? But it's tough. And what we have learned, and this is kind of interesting, and I think this is what Jay went on to say, that sometimes Tourette's can be, there are qualities of Tourette's that can be gift-like. You have superhuman qualities. But what we find is that these mice develop habits much quicker. They have difficulty suppressing them or breaking them. What's interesting, in the beginning stages of a habit, what do we see? What we notice is that these mice are super motivated. They work really hard. If you put a Tourette mouse, quote unquote Tourette mouse, next to its unaffected sibling, and you give that mouse a reward, but let's say that there's an uncertainty to the delivery of that reward. The mouse knows that if I poke my nose in this hole, I will get that reward, but I don't know when it's going to happen, so I just got to poke. What we find is that the Tourette mice very quickly learn. They elevate their nose poking so high, the rate of nose poking, that they're not going to miss that chance to get
They
that
want that
reward.
treat.
They want
Yeah.
it. Their reward perception, the idea of what that treat is to them, it may have a higher incentive value such that they're more motivated to go for it. And they repeat that action over and over again. And you see that very quickly. So they show elevated motivation. They show higher degrees of motor responding. Now, when you tabulate who wins the most rewards, the Tourette mice always win the most rewards. They always outpush the normal mice. They have that intrinsic motivation. But
But they stop those goals.
there's a trade-off for everything in life. That trade-off that Tim Howard might say gives them that extra motivation or that determination. A lot of this is fueled by that neuromodulator dopamine. Dopamine is very big. It powers our motivation. It determines our ability to perceive a reward. It also invigorates our motor system. You might have more dopamine. It might make you more motivated. It might make you more likely to repeat things, for example, or just to consolidate, as we say, motor movements. What it also might do is potentiate systems in our brain that learn to associate a sensory cue with an action, such that when we put our mouse back in our chambers, they have these specific chambers, and when they go inside, they see the nose port. They see the chamber. These are the sensory cues that the mouse has learned to associate. I'm in this assay. I'm supposed to nose poke, as we say in our lab, start poking. These sensory motor transformations are potentiated in these mice. So the same dopamine, it's working to help the motivation. It's working on the reward system. It's probably always also working on strengthening stimulus response associations that ultimately elicit a motor behavior, such that
That's exciting.
Yeah, you learn quicker. But these were ideas that were postulated from a single human study. And I should add that those that were on dopamine blocking medications did not show this habit phenotype in humans.
There you go.
Yeah. So what have we learned? Our mice are actually mimicking this theory of Tourette's. And we should add cognitive behavioral intervention therapy, habit reversal therapy.
That's the main gold standard treatment now for tics and as a therapeutic.
Right. And through these mice, we're starting to learn there's a reason for that. And that's because the habit activating centers, the parts of your brain that control habit-like behaviors, are affected in Tourette's. Through the mice, we're now starting to say, what are those particular neurons? And what we can actually do, and this is exciting, the student in my lab, the grad student who has Tourette's, who works on these mice, and in fact has developed nose poking tics by watching the mice behave.
We have the first species to species example of tick contagion. Wow.
She's mouse trained.
You should document that. Wow.
Yeah, no, she said she went from the head twitches to nose poking.
Wow.
And so, and again, that just really shows you that these traits can be learned. But what she's doing now with her student who also has Tourette's, we have multiple individuals in the lab who not only work on these mice, but they've lived with it their whole life. What they can do now is they can use these new technologies. We can put in sensors that allow us to detect dopamine live. And we can put a fiber optic cable into the brains of these mice. And while they behave, we can actually image I should say millisecond by millisecond, the dopamine changes in the brain. And we can start ascribing these changes to the particular behaviors while they're forming a habit. So,
that's huge.
It's huge. And then we can...
that relates to the bigger question in people. Why do we do what we do?
Yeah, this is some biofeedback. I mean, I would love to know what's going on in my brain, like while or especially when I'm getting a new tick or when I'm ticking. Like, I would love to have that data. And
But if your tick is just an exaggeration of a normal behavior, in fact, we have the ability using these mice to understand the normal behaviors and how they work.
We can use the same technologies to ask, okay, what do we see in a non-medicated mouse? What do we see in a medicated mouse? How are these drugs working to modulate these neurosignatures that we're recording? Really, what I'm trying to say here is that don't be disappointed that a mouse doesn't vocalize or have an actual tick.
Well, we don't know that yet.
We don't know that yet.
They could have coprolalia for all we know. We're going to find out.
And I just can't hear them cursing at me. That's very possible.
How does a mouse say go F yourself?
Yeah, but
right?
I really look at it deeper than that, the neuroscientist in me. And I say, mice form habits. We can do this. We can get into the underlying basis of what a tick is, because tics are so different in everyone. Tics can be compulsive. You don't even know it's a tick sometimes. right? There could be behaviors that you don't really know are a tic. The idea is that there's an underlying basis to these, and we're going to go find it.
You definitely challenged my paradigm on that. And that makes a lot of sense. Like, what is, deeper going on when we have a tic, just as tics can vary dramatically between individuals. But it's not about mimicking that exact kind of tic that we see in people. It's about whatever's going on underneath. In case any of our listeners are concerned about these mice and they hear us talking about giving them mutations and they're having these changes, Jay, I'm reminded of something you said at the academy, that research is expensive. Research also takes a lot of time. And especially when you're doing genetic studies where you need to get multiple generations, we're looking at, okay, we gave them this mutation. Does it get passed down to their offspring? If a mouse dies in the midst of research, that's a lot of money down the drain, and all the time and those 16-hour days, Max, you mentioned.
And they do die.
So
from unrelated causes. Our mice are looked after very carefully. Every day, an animal technician looks in the cage, and we can only have 5 mice per cage maximum, and looks for any mice that appear unhealthy. And sometimes they tell us how to treat the mice. We may have to rub some ointment on or something like that. Or sometimes they tell us that mouse is not going to live and we have to get rid of it.
Wow.
Of course, each cage costs something like $1.30 or $1.40 a day. And we have like 300 cages at this point.
Whoa.
That's every day.
There's a reason why we use mice, and that is because they're the lowest vertebrate species that can mimic the human brain and the human physiology. There's certainly ethical reasons why we don't use monkeys or why we don't use other mammals, but mice can reproduce our physiology to a degree. And also, mice, as many who might have a mouse problem in their house, they can have litters, 8 to 10 pups. Every time a mouse gives a litter, you might have 5 newborns that carry that specific mutation.
Mice are the cheapest vertebrate to keep in numbers. There's nothing cheaper. Rats are much bigger and they're much more expensive. People have been using mice well over 100 years now. And there's, I don't know how many zillion papers involving mice, but it's a very well studied, well documented. We know its anatomy. We know a bit about its behavior. We know its genetics.
I remember you saying, Jay, that you found we treat mice, our lab mice, better than we treat people. That
on a worldwide basis,
on a societal level, like we care about our research. They're getting the best food, they're getting exercise, they're getting, like, when they're sick, your research assistants are rubbing the lotion on them. I mean,
well, when they're sick, we actually have a veterinarian who comes in and looks at them. It's not technicians who make decisions on how to treat mice; it's vets.
They get specialized care.
Mice in the wild don't live very long. Because we treat them so well, they actually live longer. And our mice will live 1.5 to 2 years.
So we're getting to the end of time here. I want to say briefly, if there's any of our listeners who would like to take part in the Genetics Repository, have their information stored. Max and Jay, what would you recommend? Where do they go to get started with that process?
Our contact information is widely available on the web.
We'll put it up with the podcast as well, so they can get it there too.
Reach out, contact us through NJCTS or through our websites, and we'd be happy to connect you.
Thank you both so much. It's been a riveting conversation, and I wish you both a wonderful evening.
Yeah, no, thank you very much, Michael. It's a pleasure.
Thank you for listening to The Uptick, brought to you by the New Jersey Center for Tourette Syndrome and Associated Disorders, empowering you to stretch the boundaries to live your best life. The NJ Center for Tourette Syndrome and Associated Disorders, NJCTS, its directors and employees assume no responsibility for the accuracy, completeness, objectivity, or usefulness of the information presented on this podcast. We do not endorse any recommendation or opinion made by any guest, nor do we advocate any treatment.